Hello friends, welcome to my course on Microbial Biotechnology. We are in module 7. Today we will be discussing about biopesticides and integrated pest management. Few concepts which we will be revisiting here has already been discussed in the earlier lectures. So, we have 3 sections over here. We will tell about these sections details as we come across them. So, first we will have an introduction and comparison of the advantage of synthetic pesticides and biopesticides, then classification of biopesticides, natural enemies, then plant incorporated protectants, and then biochemical metabolites and botanical secondary metabolites, mode of action of semiochemicals. So, biopesticides are derived from natural substances including plants, microbes and nanoparticles of biological origin, making them a sustainable pest control option. They offer an alternative to synthetic pesticides which can harm the environment and human health. They are generally cheaper, environmentally friendly, and have specific actions reducing the risk of harmful residues. They can be sourced from various natural materials including microbial metabolites and plant extracts. For the purpose of integrated pest management, we will be discussing a little bit of the plant extracts as well, but our focus will be mostly on the role of microbes. So, if we compare the advantage of synthetic pesticides and biopesticides, we see that biopesticides are having more advantage because they are sustainable, cost effective. We can have them from renewable sources and they are eco-friendly. But synthetic pesticides are readily available. They are broad spectrum and they are very rapid in their action. But from the point of view of environmental health and human health, the advantage of biopesticides outweigh those of the synthetic pesticides. So, we can divide biopesticides into different classes like natural enemies, predators, parasitoids, which we discussed already in the earlier class. Then we have plant incorporated products, PIPs. These are genetically modified plants that express toxic proteins against insects like the BT cotton. Then you have biochemicals, botanicals, mostly derived from plants. Then we have also biochemicals like secondary metabolites from plants as well as microbes, the semiochemicals, spinosins from actinomycetes, etc. Then we have microbial pesticides where we use the whole microorganism like fungi, bacteria, and then the virus, which of course is not a microorganism, and then protozoa and also nematode. We will be discussing about all these and to some extent a little bit about the PIPs. So, natural enemies, we can just have a small discussion. These are organisms that naturally control pest populations. They play a crucial role in maintaining the ecological balance by keeping the pest population in check. Predators are organisms that hunt and consume pest organisms during their lifetime. They are essential in biopesticide strategies for controlling various agricultural pests. Then we have parasitoids. These are organisms that lay their eggs on or in a host insect. The developing larvae feed on the host, eventually killing it. Parasitoids are highly specific, making them effective in targeted pest control. Then we have the plant incorporated protectants, which are pesticidal substances produced by plants as a result of genetic engineering. These substances are intended to protect the plants from pests by incorporating specific genes into the plant's genome. PIPs are created by transferring specific genes from other organisms like bacteria into the plant. These genes enable the plant to produce pesticidal substance, typically a protein that targets and kills specific pests. So, what is the mechanism of action of these PIPs? For example, in BT crops, the Bacillus thuringiensis gene encodes a protein which, when ingested by certain insect larvae, binds to receptors in the insect's gut, causing cell lysis and death. The target of these kind of toxins are very, very specific, which ensures that only particular pests are affected, minimizing harm to non-target organisms including beneficial insects. A common example is the incorporation of genes from the bacterium Bacillus thuringiensis into crops like cotton, corn, soybean, which allow these plants to produce a protein that is toxic to specific insect pests. So, these is the bacteria from which this Bt gene is transferred to a target plant, and when these insect larvae eat the leaf of this transgenic plant, the Bt protein is ingested, and which is first released as a protoxin, and as it moves down the gut, it becomes activated due to the action of enzymes, and these are released, peptides are released, which bind to the receptors in the gut, which increase the permeability because of the pore formation, and thereby there is imbalance of fluid, and the insect feel like overfed and do not feed, therefore it starves, leading to its death. So, biochemical pesticides on the other hand are naturally occurring substances or synthetic analogues that control pest through non-toxic mechanisms. Unlike conventional pesticides, which typically kill pests directly, biochemical biopesticides affect the behavior, growth or development of pests, making them an eco-friendly alternative in the pest management. For example, we have the botanicals like neem from Azadirachta indica, and then we have garlic extract and so on. We will not spend much time on these botanicals because our focus is mostly on secondary metabolites produced by microorganisms like Bacillus thuringiensis, for example semiochemicals, then spinosins by actinomycetes, and strobilurin by wild mushrooms, etc. Botanical extracts are plant derived substances used as biopesticides to manage pests including insect, fungi, bacteria and weeds. These extracts are typically made by isolating and concentrating the bioactive compounds from plants with pesticide properties. Botanical extracts are valued in sustainable agriculture for their natural origin, biodegradability and reduced environmental impact compared to synthetic pesticides. And these are some of the famous examples like azadirachtin, which is obtained from neem, particularly the neem seeds. Then this is pyrethrum from the chrysanthemum flower. We have these bioactive compounds, the pyrethrins. These targets mosquito flies, moths and various other insects. They particularly affect the nervous system of the insects, leading to paralysis and death. And then we have garlic extract, which have sulfur compounds like allicin, and these are fungicidal and nematicidal. And then it can target aphids, caterpillars, fungi and nematodes. They basically disrupt feeding and reproductive processes and inhibit fungal growth. Then we have capsaicin, which is obtained from chili. They are used as insect and mammal repellent, and then target insects, rodents. They cause irritation and deterrence due to the pungency. Then we have quassia, which produces quasinoids, and they are used as insecticides against aphids, caterpillars, beetles. They disrupt the insect feeding and the digestion. Another class of biochemicals are the secondary metabolites, which serve as essential biopesticides derived from plants and other sources. These compounds play crucial role in pest management due to their diverse modes of action targeting various species. Semiochemicals, insects and plants communicate using special chemicals called semiochemicals. These are substances that carry messages. For instance, a flower might release a sweet smell to attract bees. When insects detect these semiochemicals, they change their behavior based on the information they receive. For example, a male insect might follow the scent of a female's pheromone to find his mate. Similarly, insects can find plants that are good for laying eggs or feeding by following chemical signals. The mode of action of semiochemicals takes place in different ways. It may disrupt the mating cycle, where female sex pheromones are used by insects to communicate. For example, in vineyards, synthetic grapevine moth pheromones can be sprayed to confuse male grapevine moth, preventing them from finding females to mate with. This reduces the moth population by limiting reproduction, similar to putting up a no entry sign for the males. Secondly, it may attract insects into traps where semiochemicals are used to lure insects with an attractive scent. For instance, fruit growers use pheromone coddlemone to attract and capture codling moths, which are pest of apples and pears. The trap use a small amount of pesticide to kill the moths, making these method more environmentally friendly, like catching a mouse with a small piece of cheese instead of spreading it everywhere. Then thirdly, they naturally repel insects. Semiochemicals repellents can make healthy plants smell decayed, deterring pests. For example, in forestry, methyl jasmonate, a plant signaling compound, is used to repel mountain pine beetles by making trees seem unhealthy. These protect trees without harming other wildlife, similar to how camouflage helps animals avoid the predators. Let us now discuss about the secondary metabolites of Bacillus thuringiensis, which is a type of bacteria that produces special substances to fight against pests and other harmful organisms. This is apart from the cryoprotein or endotoxin that we discussed earlier in this lecture. Basically, they produce certain secondary metabolites, for example, bacteriocins. These are antimicrobial proteins or peptides produced by bacteria to kill or inhibit the growth of other usually closely related bacterial species. They are not insecticidal proteins, but some bacteriocins can have insecticidal activity or contribute to a bacterium's insecticidal effect when combined with other factors like the cryoproteins. These bacteriocins work by interacting with the membranes, such as the outer layers of target bacteria. These interactions can disrupt the bacterial cell membrane, leading to leakage of cellular content, disruption of cellular functions, and ultimately the death of the bacterial cell. The effectiveness of bacteriocins depend on their specific structure, which can vary widely, such as being a peptide or a protein, and their mechanism of action. Different bacteriocins may exhibit varying degrees of effectiveness against different bacterial strains. For example, some may target gram-positive bacteria, while others may be more effective against gram-negative bacteria, depending on the composition of the bacterial membranes and the bacteriocins' mode of action. Bacillus thuringiensis produce several bacteriocins, which are antimicrobial peptides that help the bacterium compete with other microorganisms. Some of the notable ones are thuricin. This is discovered in 1989, and it is one of the first bacteriocin identified from Bacillus thuringiensis. Thuricin exhibits antimicrobial activity against various gram-positive bacteria. Then there is tocaisin, which is discovered in 1997. This is a antibiotic bacteriocin with microbial properties, antimicrobial properties, and produced by certain Bacillus thuringiensis strain. Then there is crustacean, which was identified in 2000, basically a cyclic antimicrobial peptide with activity against a broad range of gram-positive bacteria. Then there is thuricin 7, discovered in 2001, which is a more potent variant of thuricin, having broader antimicrobial spectrum or activity, and is particularly effective against gram-positive bacteria. Then there is another variant, thuricin S, identified in 2007, which has unique antimicrobial properties that differentiate it from the other forms of thuricin. Then there are others like thuricin 439A and 439B, identified in 2003. They are closely related bacteriocins which demonstrate activity against gram-positive bacteria. Then entomycin, which was discovered in 2003, has been reported to have insecticidal properties. Then there is bacthuricin 4, discovered in 2005, which is an antimicrobial peptide with activity against the gram-positive bacteria. Then there is thuricin CD, discovered in 2010, which is a potent bacteriocin with strong antimicrobial effects. So, we see that Bacillus thuringiense offers so many different kind of bacteriocins, and some of these are effective against gram positive bacteria, and some are also having insecticidal properties. Let us discuss about secondary metabolites from actinomycetes like spinosins. These are a novel class of highly potent natural insecticides discovered in the culture of the soil dwelling actinomycete, Saccharopolyspora spinosa. This bacterium produces spinosine A and spinosine D, which are the active insecticidal components in the commercial product known as spinosad. The name spinosad we can see here in this image, one spinosine A and spinosine D is derived by the combination of spinosine A and spinosine D. The first one is approximately 85% and the second one is around 15%. So, how does spinosad, which contains 85% spinosine A and spinosine D, kill the target insect? They exhibit unique mode of action by targeting the nicotinic acetylcholine receptors in the insects. These receptors are involved in nerve signal transmission. When spinosad binds to these receptors, it causes continuous stimulation of the nervous system, leading to the overstimulation of neurons. This results in muscle spasms, tremors, and eventually paralysis of the insect. In addition to its action on the nicotinic acetylcholine receptors, spinosad also affect gamma aminobutyric acid receptors or the GABA receptors, which are responsible for inhibiting neural activity. By inhibiting GABA receptors, spinosad disrupts the normal inhibitory control over neuronal firing, further contributing to nervous system dysfunction and the rapid paralysis of the insect. The dual action of both the nicotinic acetylcholine receptors and GABA receptors makes spinosad particularly effective against the wide range of phytophagous insects, including pests such as thrips, leaf spiners, and caterpillars. So, spinosad is biologically active against several key pests, especially lepidopteran insects, including tobacco bollworm, cotton bollworm, American bollworm, and army worm, Spodoptera litura. Secondary metabolites produced from wild mushrooms like Strobilurus tenacellus, so which produces strobilurin. These strobilurins represent a new generation of fungicides derived from this particular wild mushroom. Particularly strobilurin A and B, these are the first isolated such compounds from the wild mushroom Strobilurus. How does these strobilurins act? They inhibit mitochondrial respiration in fungi, both higher and lower fungi, disrupting energy production. These fungicides exhibit translamellar energy production, moving across the leaf blade to protect both surfaces, even if only one side is treated. Some like azoxystrobin also move systematically within the plant, offering comprehensive protection. So, commercially these fungicides are now available in various formulations. So, like some of these are the azoxystrobin, Quadris, Abound, Amister, and Heritage, and also trifluoxystrobin, Flint, Stratego, and Compass. And these are having efficacy against various plant diseases, like for example, the studies on pearl millet downy mildew showed that azoxystrobin provide up to 91% protection, which is quite high. Now, let us discuss about the production of formulations of biopesticides from metabolites. So, mostly we depend on fermentation, where we start with inoculum preparation. We culture a small quantity of microorganism in a laboratory setting to generate a sufficient quantity for large fermenters. So, we go for scaling up, gradually increasing the scale from sec flux to pilot plants and then to full scale fermenters. Continuous monitoring of the growth parameters like optical density, pH, temperature and metabolic production through the sampling is very important to understand the growth dynamics. So, next comes the formulation of these particular production. So, we may start with isolation and screening from the natural sources, and then also the characterization, and then flask cultivation, and then lab scale cultivation, and then finally industrial scale, large scale production. The next stage important is the formulation stage. So, once we harvest the biomass or the microorganisms and the metabolites at optimal time based on the growth curve analysis, we go for concentration and purification using techniques such as centrifugation, filtration, chromatography to concentrate and purify the metabolites, and we will go for formulation of granules or powder or liquid or solid formulation. So, we combine purified metabolites with carriers like adjuvants or surfactants to ensure stability, effectiveness and ease of application. We also go for testing and quality control, evaluating the efficacy and safety of the formulated biopesticide through laboratory and field trials, followed by quality control testing to ensure consistency and reliability. Then of course, we will go for the various kinds of applications which can be foliar or which can be soil application, and then field trials are very important, and constant monitoring very important in this entire process. Now, let us go into section 3 where we will discuss various microbial biopesticides, where microbes are directly used as biopesticides. Here we were mostly using metabolites as biopesticides. So, we can use bacteria like Bacillus thuringiensis, which produces harmful toxins against specific insects, or you may use fungi such as Bovariata bassiana that infect insects leading to their death. Then we may use insect specific viruses such as the nucleopolyhedroviruses, or we may use entomopathogenic nematodes, which are microscopic roundworms and that parasite and kill the insects. So, let us start with bacteria. Bacterial biopesticides are single celled organisms that produce toxins or other substances for killing the pest. They are grown in large scale to produce pesticidal formulation. So, we may have different categories of bacteria used as pesticides, mainly 4 types. So, among these, spore forming bacteria are the most popular for making biopesticides because they are very effective. So, we have a classification of bacteria, bacterial biopesticides. Some are endospore forming bacteria, some are non-endospore forming bacteria, for example, Serratia. So, some of these endospore forming bacteria may be obligate like Bacillus popili, or some are facultative, some are crystal forming like the Bacillus thuringiensis, and others are non-crystal forming like Bacillus cereus. So, then we have these entomopathogenic fungi which are used as biopesticides. These are known as the fungal pesticides or micro-insecticides, which use fungi to target and control insect pests. They start their infection when tiny particles called spores touch an insect. The conditions for infections are they prefer moderate temperatures, not too hot or too cold, and high humidity. Under these conditions, the spores can start to grow. Some of the examples of entomopathogenic fungi are Bovariata bassiana, Metarrhizum anisopli, and then Trichoderma hargenium. Then we have certain spores which infect insects, which are produced by the entomopathogenic fungi. Once the spores land on an insect, they begin to grow and use special chemicals particularly enzymes, to break through the insect's tough outer layer, the cuticle. They can also push their way in using mechanical pressure, which is like using force to get through a barrier. So, the first step is attachment and germination. Conidia or spores of entomopathogenic fungi attached to the surface of an insect host. The fungal spores then germinate, forming germ tubes that penetrate the insect's exoskeleton. Then there is the penetration. So, this is the cuticle of, you can see over here. So, and here is the hemolymph, and then these are the proliferating hyphal bodies, the conidium, which forms the apressorium, and then it slowly penetrates the cuticle. Once inside, the fungus grow and spreads throughout the insect's body, forming hyphal bodies. The fungus colonizes internal tissues and organs, causing physiological disruption. It also produces toxins. These entomopathogenic fungi produce bioactive secondary metabolites such as destruxin, buvericin, and other toxins that contribute to the insect's death by disrupting cellular processes and causing systemic damage. Then finally, there is host death and spore production. The infected insect dies typically within 3 to 5 days after death. The fungus continues to grow and produce new conidial spores on the exterior of the host, which are released into the environment to infect other insects. Let us now discuss about entomopathogenic viruses or EPVs. They kill insects that have gained attention for pest management. Although tested since the early 1900s, the first virus based insecticide was registered in the USA in 1970 to combat the cotton bollworm. Research continues to identify and evaluate new viruses for pest control. Viruses of a few families are known to infect insects, but only those belonging to the highly specialized family Baculoviridae have been used as biopesticides. So, we have two types of Baculoviridae under the entomopathogenic viruses. One is the U. baculovirini, another is the Nudi baculovirini. So, these has U has occluded virus particles and Nudi has non-occluded virus particles. The examples of U. baculovirini contains nuclear polyhedrosis virus NPV and granulosis viruses. So, some of the examples of EPV and their target pest. So, we have these SE NPV that is targeted against the beta armyworm, and we have AZ NPV which targets Heliothis or Helicoverpa, then CP NPV Cotling moth, AG NPV velvet bean caterpillar, then AC NPV Autographa californica, and SL NPV which targets Spodoptera litura. So, what is the mode of action of these entomopathogenic viruses? The virus exists as polyhedra or tiny protective structures. When caterpillar eat plant, they consume these polyhedra. The polyhedra are ingested by the insects as they feed on plants. Once inside the caterpillar, the polyhedra reach the midgut. The alkaline midgut dissolves the polyhedra, releasing the virus inside. So, this is released in the alkaline environment of the midgut, and primary infection of the insect cell will follow. A part of the ODV, occlusion derived virus, is freed, which infects the cell lining, the midgut, and this will also further cause secondary infection and accumulation of the polyhedra, and finally the death of the host. So, the virus will start replicating into new viruses called budded viruses, and the infection will spread to other tissues. It will leave the midgut cells and spread to other parts of the caterpillar's body. And there may be also horizontal transmission. The virus spreads from infected to healthy caterpillars. due to accumulation of polyhedra, disintegration of larval body, and the release of the polyhedra. Let us now discuss about another important microbial biopesticide where we use entomopathogenic nematodes. These are microscopic worms that live in the soil. These worms are natural enemies of many harmful insects, meaning they can help reduce the number of pests without using harmful chemicals. There are different types of these nematodes, mainly from two groups, Heterorhabditis and Steinernema. These groups have several species, S. feltia, then Heterorhabditis bacteriophora and H. megidis. These are also available commercially, so we can source them from suppliers to control insect pests in the soil. One important aspect is the safety of these entomopathogenic nematodes. They are safe for other living organisms. They do not harm animals like pets or humans, plants or other beneficial organisms in the environment. This makes them an eco-friendly option for pest control, which is very important for sustainable farming. So, what is the mode of action of these entomopathogenic nematodes? The young infective stages of these nematodes are very active. They search for insect hosts to enter. They can enter inside insects through natural openings in the insect's body like the mouth, spiracles, anus, intersegmental membrane. Once the nematodes enter the insect, they release bacteria, which are symbiotic bacteria, which they carry with them. The bacteria cause infection in the insect, leading to bacterial septicemia, which is when the bacteria spread throughout the body and can cause death within 1 to 2 days. This is a very fast process. So, the infective juveniles enter by natural openings or cuticles, the bacteria is released, and then the adults of the nematodes will develop, they will produce progeny 2 to 3 generations, and then when the resources are depleted, they will be released from the cadaver. What are the bacteria that are carried by these nematodes? For example, Heterorhabditis carry Photorhabdus species, then Steinernema carries Genorhabdus bacteria. These are crucial for the nematodes to effectively kill their insect hosts. Let us now discuss about another important microbial biopesticide, the protozoa, which are single-celled eukaryotic organisms, and these are used as biological pest control agents due to their ability to infect and kill various insect pests. However, the practical application in biocontrol is very sporadic. Bacteria, fungi and viruses used as biopesticides are often easier to produce and have a broader spectrum of activity. These alternatives are more widely studied, better understood, and already integrated into pest management systems, making it harder for protozoan biopesticide to compete in the market. One of the most commonly used protozoan biopesticides in spite of these is the Nosema species. So, we have the Nosema locustae, which is known to infect grasshoppers. This is important because it is the only protozoan species that has been successfully used in farming to control grasshopper populations. Then we have Nosema bombycis, which affects silkworms and causes a disease called pebrine. This disease has been around for a long time, especially in places like Europe, North America and Asia since the mid 1800s. Silkworm, being an insect of economic importance, are not considered a pest, and in fact efforts are there to control Nosema bombycis infection. How do the protozoans infect insects? Protozoans can produce tiny particles called spores. They are like seeds that can spread the infection. When an insect eats these spores, they enter the insect's body and start to grow in a part of the digestive system called the midgut. Once inside, the spores begin to develop and break out of their protective covering. Then they invade the insect cells, causing a lot of damage to the insect's organs and the tissues. After causing damage, the infected tissues can produce more spores. These new spores can then be expelled from the infected insect and can be eaten by other healthy insects, spreading the disease further. In nature, some insects that eat other insects called predators, and certain types of webs also known as parasitoids can help spread the protozoan disease. They act like carriers, moving the spores from one insect to another, which helps the disease to spread even more. Let us now discuss about the biopesticides from microalgae. Some types of microalgae produce biologically active compounds. These compounds can fight off harmful bacteria and fungi, which are types of pathogens that make plants sick. Because of these, compounds can be used as biopesticides to protect crops. Microalgal biomass can be used instead of the chemical pesticides. So, what are the different types of biopesticides from microalgae that has shown the ability to act as biopesticides? So, some of these are the nostoc species, which is a type of filamentous cyanobacterium. Is a long, thin, thread-shaped microbacteria or microalgae. It can help protect plants from certain diseases. Then we have Anabaena variabilis. This is being explored as a possible biofertilizer and biopesticide for growing rice sustainably, helping to improve crop yields without the negative effect of chemicals. Then we have Chlamydopodium fusiforme, which is a single cell green algae that can also help fight off plant pathogens. In spite of all the good things we have discussed, there are certain limitations which are there associated with the biopesticides. For example, we have challenges of storage. They may become unviable, particularly the living organisms which we use as biopesticides need specialized type of storage. Then another problem is of their high specificity. Not all of them are having broad spectrum activity. So, this limits their effectiveness. But when it comes to single species infection of a agricultural field, then they are actually very beneficial. And then there is always the challenge of culturing in large quantities or scaling up, and then also the timely application may sometimes be a challenge. We may not be able to get the sufficient amount by culturing due to the time that is required for producing in large quantities, or even the supply chain may be sometimes not very logistically sound, and it requires many additional control measures. For example, if we are using live microbes or like fungi, bacteria, etc. as biopesticides, and if the field is already contaminated with broad spectrum pesticides which are harmful to these organisms, application of the biopesticides may fail. So, we need a total organic system where these pesticides biopesticides can be highly effective. So, let us once again revisit the concept of integrated pest management that combines different ways to manage pests, which are unwanted insect or animals or microorganisms that can damage crops. Instead of relying on just one method, IPM uses a mix of techniques to keep plants healthy. So, we use all the different kinds of pest control methods available in IPM. So, what are the benefits of IPM? Number one, it provides environmental protection, it is highly profitable, it also results in societal benefits, and there is a risk reduction. However, there are challenges in adopting IPM because not many farmers use them due to reasons like lack of awareness, then user preference, using synthetic peptides or due to different reasons, maybe marketing being one of them. And then technology inaccessibility. Many times IPM is mostly like a package, and it needs to be taken to the field by extension workers, and certain farmers may not have access to those kind of technological support, and also the policy and culture impacts the adoption of IPM in a particular region or a locality. So, what are the different principles of integrated pest management? It relies on various principles like natural control, sampling economic levels, then insect biology and ecology. Natural control involves enhancing naturally existing factors that suppress insect pests by creating an ecosystem less favorable for their growth. This can be achievable by managing the environment to limit pest population growth and by promoting beneficial insects that help keep harmful pests below economically damaging levels. In the sampling of economic levels, we go for determining economic threshold levels to guide control measures, particularly chemical ones. By establishing dynamic economic threshold levels, insecticide use can be delayed, allowing for the optimal use of alternative methods like promoting beneficial insects. Insect biology and ecology involves understanding the biology and ecology of all species in an ecosystem to identify natural control mechanisms. This knowledge helps assess each species' role and damage caused by pests, forming the basis for planning and implementing effective control strategies. So, this figure provides a general step of IPM program development. This is taken from the European Union draft guidance document for IPM published in 2008. So, the first step starts with prevention and suppression, principle number 1, principle number 2, constant monitoring, number 3, decision making based on scientific studies like sampling economic levels and also understanding biology and ecology of all species in an ecosystem. So, in principle 1 involves the combination of tactics into preventable strategies. Principle 2 monitoring involves observation, forecast and diagnostic, and principle 3 involves use of thresholds, use of multiple criteria. Then we have principles 4 to 7 which mostly deal with the intervention. Non-chemical methods are the most preferred, then using agents which are having least side effects, then reduced pesticide use and anti-resistance strategy. Biopesticides may also develop some kind of a resistance in the long run, so that also has to be taken into account. And finally, the principle of evaluation, which involves the assessment of the entire process and the adoption of new standards. And these principles are to be followed in a sequential manner for efficient integrated pest management. So, with this we come to end of this lecture. Thank you for your patient hearing.